Now, the next step in the protein synthesis is known as the translation. Translation. What is translation? Translation is the process in which the information found in mRNA is actually translated to protein. So mRNA is a sequence of base pairs. Remember, mRNA is a sequence of base pairs. This now should be taken into a sequence of amino acids. So sequence of base pairs to sequence of amino acids. The key player in this, it will be tRNA. The key player in this process will be tRNA, and this translation occurs at ribosome. Now, remember what helps in the translation between sequence of base pairs? So we have sequence of base pairs, 1 base pair, 2 base pair, 3 base pairs. Now this should be translated into sequence of amino acids. What helps in that? Remember the 3-letter codes that we talk about, the mRNA codons that we talked about. So if the 3-letter code was a UUG, for example, UUG, this translates to leucine. If the 3-letter codon was UAU, then this translates to tyrosine. So these 3-letter codons will actually come into picture at this point. We said that the synthesis takes place in ribosome. What is a ribosome? Ribosomes are proteins. They have 2 units. A smaller subunit, they are divided into 2 units. A smaller subunit and a larger subunit. So in the first part of translation, which is known as the initiation, the mRNA will come and bind to the ribosome smaller subunit. So let's say this is what the mRNA looks like. This strand, this is the mRNA strand, okay, with 3-letter codons. So let's say one that I want to look at first would be your AUG, for example. AUG. So, and then I'll move on to the others as I go through. So, and just to make it a little bit easier, I'm going to make the 3-letter codes like this. So mRNA binds, and this is your first 3-letter code, so AUG. So AUG. So again, in the translation, the first step is initiation, where the mRNA binds to the small subunit of the ribosome, and the corresponding transfer RNA lines up. So let's say this is your transfer RNA, and I'll just use a different color. So this is your transfer RNA. Okay, now remember the anticodon on transfer RNA. Now this has a corresponding 3-letter sequence here, which is the anticodon, and this would be U A C, right? This would be U A C. So this tRNA has also been activated. So this tRNA has also been activated. What does that mean? This tRNA corresponds to a amino acid. So remember there was an acceptor codon here, an acceptor stem. There was an acceptor stem here and that had an OH. So what is the activation of tRNA? A enzyme called amino acyl tRNA synthase. I'm going to write it down here. An enzyme known as amino acyl tRNA synthetase. Attached an amino acid to this acceptor stem, and in this case I'm going to say this amino acid is methionine. Okay, so this tRNA has been activated. What is the meaning of activation? An enzyme known as amino acetyl tRNA synthase attached an amino acid to the acceptor stem of this particular tRNA, right? Now if you notice, these are complementary. The UAC, and I'll just show you using a different color just because I want to make it easier. These strands are complementary. UAC is complementary to AUG right here. Okay, and I'm not showing the attachment here, but it's actually attached. Now, as a next step, what happens is the larger subunit completes the initiation complex. What's the purpose of this larger subunit? What it does is every time a tRNA comes in and binds, the larger subunit can actually the ribosome actually travels through, or the mRNA travels through the ribosome. So the next thing what happens is the second tRNA binds here. So let's say our second sequence here is UUA. Okay, let's say the next sequence here is UUA. So another tRNA binds. Okay, and I'm just going to show not the entire thing. Another tRNA binds with a complementary sequence which would be AAU, and of course this would be specific to another amino acid, and in this case the amino acid would be leucine. Now what happens is that this mRNA now moves out. So what happens to this mRNA now? So this whole site moves forward. That means it will come to a position where it's going to look like this. So this is your larger subunit. Your methionine now moves back, moves one step forward. Your leucine comes here, and to the binding site, which is known as the acceptor site, in the larger subunit, the third amino acid tRNA comes into place. Then after that it moves one step forward. So in that case what happens? This moves forward, this moves forward, this moves forward, and then in this place the next tRNA comes into place based on the amino acid sequence. And as this process keeps taking place, what you also have to remember is that these amino acids that are present here will keep forming peptide bonds with each other. This peptide bond formation is also catalyzed by the ribosome itself. The ribosome catalyzes the peptide bond formation as well. So every time a new tRNA comes in, the ribosome larger unit slides over, and so the peptide bond keeps forming, and this process is known as elongation. Elongation. So this process keeps repeating, and we come to a stage where the mRNA is bound to the smaller unit of the ribosome, and every given instant there are about two tRNA bound. So I'm going to show the tRNA in blue color. tRNA corresponding to a certain protein. This corresponds to a certain protein, and these corresponds to a certain amino acid, and these were the amino acids that were brought in before, and this was the peptide bond that was formed. And of course there was the larger subunit of the ribosome, and then finally at one point they come to a situation where they come across a sequence UGA. They come to a sequence UGA. What is UGA? UGA is a termination codon. Termination codon. So this is the signal for termination of peptide synthesis. So then what happens is that this larger subunit and the smaller subunit disintegrate, the complex disintegrates, and the amino acid sequence is released.